We are ready to move on to the next scientific session speaker, Debra Van Egeren. Oh, great, yes, Kristen has made her a co-host. So Debra, you should be able to share your screen when you are ready.
Perfect, sounds good. All right. Let me arrange myself here. Great. So yeah, thanks for the opportunity to present here. I'm excited to share a body of work that is exploring the role of our current vaccine-centric COVID control policies on ongoing viral evolution during this pandemic. So in case you missed it, President Biden actually recently declared the COVID-19 pandemic over, which is interesting, but you may not quite remember that the pandemic has been over other times as well, it turns out. And there does begin to be a bit of a pattern that emerges actually, where we see a slowdown in terms of the new case counts, and then somebody declares that the pandemic's winding down, and then we see a resurgence in cases. We've seen this a couple times in 2021, and there's no great reason to believe that this nadir in cases will last for very long either. As you may well know, these waves of disease are generally driven by new SARS-CoV-2 variants. And as the virus has evolved throughout the trajectory of the past couple of years, our understanding as a scientific community has also evolved as to the role of evolution in the overall trajectory of the pandemic. So initially, I think there was some confusion or doubt as to how much of a role mutation and selection we were going to play during the pandemic. But as we kind of experienced I think the Delta wave was the first time we really thought as a community that understood as a community fully the impact of these new variants on the trajectory of the COVID-19 pandemic. And this trend has only continued with the recent Omicron cases. And now our group actually had thought very early in the pandemic that evolution would play quite a large role in its overall trajectory. Some of the first signs that we noted were that there was quite a bit of immunodominance of specific epitopes on the spike protein, and that combined with the relatively high mutation rate of the virus, as well as its evolutionary plasticity, suggested that we might possibly see rapid evolutionary escape, especially from things like antibody-driven therapeutics or prophylactics, or vaccines or natural immunity. We also noted quite early on that we recognized that within host evolution was also going to probably play a reasonably big role in the emergence of new SARS-CoV-2 variants, particularly in patients that have prolonged active infections with viral replication. Now, unfortunately, even after the deployment of vaccines, we didn't see a slowdown in the emergence of these new variants. And despite the fact that these vaccines, at least for now, are quite effective against severe disease and death, our group did predict that even under endemic steady state conditions where we wouldn't see much viral evolution going on, we'd still predict quite a bit of COVID-19 related mortality that would be ongoing in the US. Now, if you layer on top of that these additional variant-driven waves of disease, you see quite a dismal picture of continuing spikes of COVID-19 associated deaths in the US that will probably proceed into the future. And we've been recently recognizing that COVID-19 is also causing long-term medical complications in folks that have recovered from the disease, including folks that were vaccinated and had recovered. This so-called long COVID is likely to continue to cause economic and human consequences into the future. So in light of these consequences and challenges of viral evolution that we've seen over the past couple of years, we sought to explore the role of vaccination and its effect on viral evolution during this pandemic. To do this, we identified several steps in the pathway to create a new variant, so several steps in the pathway to viral evolution. And we essentially, when you create a new variant, you start out with a mutation or mutations within an individual who's infected. This new variant can be selected for within that host and then be transmitted to subsequent new hosts in the population, perhaps be selected for on a population level. Now, vaccines have been suggested to possibly interfere with all of these steps in this process. And so we sought to model each one separately, starting with the thought that perhaps vaccination is able to reduce the overall active number of cases sufficiently to reduce the pool of mutations upon which evolution can act for this virus, and thereby reducing the pace of the creation of new variants in the population. So some of the earlier evolutionary theory work that we did was to explore the degree of standing viral diversity in the SARS-CoV-2 population, given the number of active cases at any given time. And it turns out that even given pretty conservative numbers of current active cases, even in light of the current vaccination state and all that, we see essentially the existence of a significant reservoir of variants within the population that exists within individuals that have 1, 2, or 3 additional point mutations. This suggests that there is significant pre-existing fodder for evolution to act, given the application of the right selective pressure at a population level, and that evolution in this case is probably not going to be mutation limited. So then we also modeled the first step in selection, and that would be selection within individual hosts during the course of a single infection. To do this, we developed a stochastic viral dynamics model within hosts to assess the differences between vaccinated and unvaccinated individuals. So given the measured viral load kinetics within vaccinated and unvaccinated individuals from a separate study, we modeled viral replication and selection of a beneficial mutant within these hosts. You can see that the data show that vaccinated individuals actually are able to clear the virus a little bit faster than unvaccinated individuals in this data set. The difference isn't huge, but there is still some difference there. And kind of almost unexpectedly, actually, this increased clearance rate in vaccinated individuals actually leads to a stronger selection pressure for beneficial variants during that phase of infection. And in fact, that leads to higher frequencies of beneficial variants within these hosts over time, as well as an increased chance that these new beneficial variants will be passed on to subsequent hosts. Suggesting that vaccination doesn't actually reduce the risk that individuals will create or pass on a new variant into the population. Next, we more specifically modeled the initial transmission steps that a new variant would undergo when they're first created within the population. To do this, we used a branching process model to investigate the transmission and survival of new variants with 2 mutations. And vaccination in this case, what it does is it can reduce the relative fitness of these new variants. And for a very effective vaccine against transmission, you can see that if you vaccinate the entire population, you don't get transmission of these new mutants. But if you have imperfect vaccine coverage, even with a vaccine that's 100% effective against transmission, you still have a very good chance that these new 2 mutant variants can survive in the population and establish. And now, in slightly more realistic cases where you don't have perfect vaccine efficacy against transmission, you can see that even in cases where you have very good vaccine coverage in the population, you can still see emergence and establishment of these new beneficial mutants, suggesting that in general, vaccination probably is not sufficient on its own to prevent the emergence of these new variants at a population level. Furthermore, we used the same modeling strategy to assess the impact of increasing vaccine coverage on the emergence of a new mutant off the back of the current COVID-19, off the back of a current COVID-19 infection wave. So essentially, you can imagine a situation where if you deploy a vaccine and try and boost coverage in the population in response to a wave, you can ask whether you're able to do that well enough to prevent another wave from appearing off of the back of that first wave. And if you can't do this, you may get into a situation where you're playing a bit of a dangerous game of whack-a-mole with these variants, where as soon as one variant comes up, you deal with it, then another variant's going to come up right on its heels. And we found that even for a vaccine that's 100% effective against transmission, for reasonably beneficial new variant, reasonably more fit new variants, you'd have to generally vaccinate at an unreasonably high rate in the population per day to really prevent the emergence of a new variant that would follow up that initial wave. And if you have a less effective vaccine, unsurprisingly, that will make your task even harder and make it, in many cases, impossible if you have a variant that's being transmitted in the population at a reasonably high rate. This suggests that a vaccine-only or vaccine-centric policy for preventing the emergence of these new mutants may not work, and you may end up in this cycle of playing catch-up with these variants. Finally, we assessed the impact of vaccination, particularly variant-specific vaccination, on selection dynamics at a population level. This is particularly salient as we roll out as a community the Omicron-specific bivalent boosters, but it's relevant quite generally. We did this by simulating an SIR model with 2 different variants circulating in the population, one dominant variant that would be targeted by the vaccine, and another emerging variant that is less well targeted by the vaccine. And now if you have a vaccine that isn't very good at targeting that new variant, as you increase that vaccine coverage, you will end up with more selection pressure for that new variant. That new variant will grow relatively faster than if there was less vaccine coverage, for instance. Therefore, vaccines that are pretty specific against the variant are likely going to encourage the development of these new variants, new immune-evasive variants, new variants that are distinct enough from the dominant variant or the vaccine-covered variant to be able to evade that prophylactic. So in summary, I've talked to you about a few different modeling studies investigating the relationship between vaccination and viral evolution in the SARS-CoV-2 pandemic. In general, unfortunately, contrary to kind of what we and probably everybody else were hoping, we didn't see sufficient reduction in the pace of our evolution for any of these specific steps in this pathway to really halt the development of new emerging mutants in the population. So that's a little bit unfortunate, and it suggests that we need to maybe have a strategy that's not vaccine only to help contain the evolutionary kind of drive of this virus. So how do we do better in terms of policy, in terms of focusing on, in terms of like our focus as a scientific community? One thing that's particularly salient, that actually probably was better established in some of our other work, is that it's really important to not only limit severe disease outcomes, but also limit transmission to as low as you can get it, to slow evolution significantly. Because this virus is like quite transmissible and mutates quite a bit. So you have to slow transmission to a pretty low level to get effective results. And this is probably going to involve having a an aggressive multi-layered strategy that includes things that we've done: masking, ventilation, surveillance testing, isolation, and particularly for individuals with longer COVID infections, to help slow the pace of evolution sufficiently. We can also think about designing vaccines and other therapeutics and prophylactics with viral evolution in mind. This might involve making sure that they can broadly target a broad spectrum of variants, including existing variants, but also possible variants in the future. And perhaps think about designing these strategies to put evolutionary pressure on the rest of the virus, not just the spike protein, because the spike protein has been shown to, mutate quite rapidly, and those mutations are often the cause of increased transmission or immune evasion in the virus. With that, I will end. Thank you all for listening, and thank you to the folks that made this work possible, both at Fractal Therapeutics and at various academic institutions. And I'm happy to take questions. Thanks.
Excellent. Thanks very much, Deborah, for very interesting and timely work. So I will open up for questions, and I see we already have Laura's raised her hand. So Laura, would you like to go ahead and unmute yourself and ask the first question?
Yeah, I have two questions. Thank you for a very thought-provoking talk. Can you go back to that slide where you show the long COVID effects? I have a question about that first. Yeah, there. That
This
one.
Yeah.
So, so the title is kind of like a little bit confusing to me. So for all of these symptoms, what you're showing is it has a ratio for people who have been vaccinated and had breakthrough infections. Is that what it is?
Yeah, so the hazard ratio is with a contemporary control group that has not been infected. So it's not like vaccine versus vaccine versus no vaccine. It's vaccine with an infection versus no infection.
Right, but that's kind of a little bit misleading, right? Because I mean, a fairer comparison will be vaccine versus no vaccine, both groups infected.
Oh, certainly. Yeah, sorry. Perhaps the title then, as you were saying, was misleading. The point was not to suggest that vaccination makes this worse, but rather that despite vaccination, folks are still seeing complications.
Right. Okay. Yeah, I definitely think that reading at the title, it almost sounds like people should not be vaccinated looking at this graph. I think the comparison is, yeah, the comparison is not really a fair comparison, because if you were to get COVID, all of those bars will be far worse, right? Even with the vaccine.
Yeah, so I actually don't remember from this paper whether—
Sorry. So yeah. If you were to have COVID and you were not vaccinated, I mean, for sure your hazard ratio for death will be maybe 10 times higher or something. So I mean, I just think the comparison of vaccine versus not getting infected is really not a fair comparison. I mean, the comparison should be between vaccine and infected and unvaccinated and infected. But
yeah, sure. That's yeah. I think they did this in that paper. I don't remember exactly what the results were, but sure, yes.
And then my second question is, in one of your contour plots, a little like when you are talking about the RTs, a little bit farther away. No, yeah, I think there. Yeah, so I was curious about RT. Like those RTs are very high to me. Like, can you talk a little bit about the actual possibility of an RT of 5? I mean,
yeah, so that's closer to maybe the R naught of wild type SARS-CoV-2, right? That's probably more on the upper end of stuff, or maybe even some of the newer variants, right? You can imagine us having that sort of RT if we took a lot of, like, basically took all of the control measures out. So that I feel like that is possible, especially if vaccine immunity has waned for a bit. To be fair, at least for the most part, RTs are a little bit closer to 1 or between 1 and 2, and less close to maybe 5 in recent
yeah, that's my point. I don't think we saw anywhere, even without any interventions, an RT above 2, even for Omicron. So I feel that, yeah, I feel definitely an RT of 5.7. I mean, I don't know what the R naught for that will be, but it will be gigantic considering pre-existing immunity and vaccinations.
Certainly.
Thank you. Very interesting talk.
Yep.
Thanks, Laura. And I see that there's a question from Chris in the chat. Debra, I'm not sure if you can.
I'm not doing a great job with chat here.
Oh, okay.
Oh yeah. Oh, here. Yeah. No, I can see it now.
Okay.
I'll just so as the question, I believe, is regarding the again, the long COVID plot here. Correct me if I'm wrong, but I think the question is if about kind of the hazard ratios here, if you're comparing uninfected individuals versus people with vaccine breakthrough infections, why would you see these outcomes in the control groups, right? And go ahead.
Not why would you see the outcomes so much as just trying to confirm a sense of what you really mean by at least one post-acute sequela is simply at least one of the below outcomes, which are reasonably common post-acute sequelae in people who have COVID, right?
Yes. Yeah.
Okay. Just a fiddly point
Sorry.
that I want to make sure I grasped.
Thanks, Chris. Any other question? Oh, yes. Another question in the chat.
Yeah, so I think the question in the chat is about validation with real world data. So yeah, I mean, it's a little tricky as an evolutionary biologist to do some of these validations on a population level. I will say that I think we did a little bit of validation in the sense that you can go back and compare the you can compare the predictions of the model to some events that occurred at the population level, like, and it's a little bit difficult because evolution is a bit unpredictable to predict the rise of like a particular variant, but we certainly did predict the rise of variants that had particular properties, and that sort of variant was likely to arise and establish itself in the population. That's kind of the limit of validation that we had. And yeah, we are currently, exploring ways to incorporate more like genomic data that's publicly available, but no word on that currently.
Thanks, Debra. So we probably have time for maybe 1 or 2 more questions. If anybody has anything else they'd like to ask. If not, we can thank Debra again for her excellent presentation.